Hello everybody, how are you all? Welcome back to Understanding Cancer Season 2 series. Today we are going to do Lecture 3, Types of Cell Signaling. So let's have a recap of what you hopefully should have understood from last week's lecture. There are 8 hallmarks of cancer developed by Hanahan and Weinberg: sustained proliferative signaling, evading growth suppressors, resisting apoptosis, enable replicative immortality, invasion and metastasis, angiogenesis, reprogramming energy metabolism, and avoiding immune destruction. To facilitate these hallmarks of cancer, there are 2 contributing factors: genomic instability and tumor-promoting inflammation, and this is what underlies most of the hallmarks of cancer. Now, the cell cycle presents how normal cells divide and grow, and there are 4 phases: gap 1, S phase, gap 2, and mitosis. There are 2 types of apoptotic pathways, and that depends on molecular scissors and enzymes called caspases. There are 2 types of apoptotic pathways: intrinsic and extrinsic. Cancer cells interact with other cells in the epithelial-mesenchymal transition in order to invade and metastasis. To have further information on all of these key points, please refer to Lecture 2. There is a supporting glossary to help understanding any of the keywords. So what will we learn today? Today we aim to learn what is cell signaling, and we're going to divide each of the types of signaling pathways with examples to help support your learning. The first type of signaling pathway is paracrine signaling. The second type is contact dependent. The third type is autocrine, and the fourth type is endocrine. A gentle reminder that this lecture consists of 5 mini lectures, and you can divide it across the 7 days and work at your own pace. Okay, you can also challenge yourself with a quiz, and hopefully this lecture should not take more than 1 hour. So a gentle reminder of the package for each of the lectures. You have key facts with diagrams by HN Designs presented in a simplified array. Okay. So within each of the lectures, you have PDF notes that has diagrams with images and key facts to help support your learning. There's also a glossary, so any keywords that you're not sure of what they mean or be able to describe or understand, then please refer to the glossary. There are also summary doodle revision posters in every lecture and quizzes to be able to test your knowledge and reflect on the knowledge that you have obtained. There is also a reference list for further reading for anybody who's interested to read further via the respected references. A special thanks to all my friends, my family, my colleagues for their support, and the respected teachers and health professions who ignited the passion of cancer within me. So what is cell signaling? Cell signaling is how cells communicate with one another and requires 3 steps: receptor activation, signal transduction, and cellular response. Now in Lecture 2, we have come across some of these, for example, within apoptosis, how the signal has been activated, then it's been transduced within inside the cell, and then you have the necessary genes being transcribed. But the aim within this series is that we're going to introduce it in today's lesson, and then we're going to divide each of these steps as a separate lecture to help you understand more in depth. So receptor activation is when you have an internal or external stimuli. So internal means inside, external means outside stimuli, which is like a signal that can influence the properties of the cell. So in order for it to be activated, it needs to bind to its specific receptor, just like a lock and key. Okay? And just as a recall, a similar case when we looked at the structure of the DNA, so the base pairs, so when they're paired complementary. The next step is signal transduction. So when the receptor has been activated, we need to transmit this signal, okay, within the cell. And this requires proteins and enzymes that help facilitate this process. Then cellular response. So there can be 3 different types of responses. The first response is regulating genes that are needed to be transcribed and translate to form the actual proteins in question. So in the previous lectures, we mentioned that in order for the genetic material to be converted into a protein, okay, it has 2 steps: transcription and translation. Okay, so once you have the template being produced within transcription, the coding within the template is then being translated in the ribosomes, okay, in order to make the specific protein in question. So the genes are transcribed within the nucleus, and the template is being translated into proteins via the ribosomes. And proteins, there's a variety of different functions, such as growth, movement, also to be able to differentiate into specialized cells, also proteins that are involved within cell death or synthesis of other proteins. Another way in how the cellular response occurs, besides regulating genes, is structural proteins. So structural proteins that can help change the shape of the cell, and also enzymes. So enzymes are a specialized type of protein, and what they do, they help to speed up the chemical reaction without being chemically changed. Okay. Now, dysregulation of the signal transduction pathway and how the signal is being transmitted throughout the cell via the proteins and enzymes, when it's not working properly or it's not been regulated appropriately, that is what underlies most of the hallmarks of cancer. And there are a variety of different types of signaling pathways, and this varies based on the target cell and also the distance travelled. So here is one of the doodle posters, okay, and that presents a signaling pathway. So the step 1 is the ligand, which is the signaling pathway or the molecule, and that complementary binds with its specific receptor. And then you have a series of different proteins and enzymes that are involved that transmits the activated signal and passes through the cell. Now, what happens is that you have a transcription factor that helps facilitate the specific gene that you want to be produced. So this takes place within the nucleus. So the transcription activator or the factor there enters the nucleus, and then it helps ignite or start off the gene transcription of that particular gene. So let's begin with paracrine signaling. So what is paracrine signaling? Paracrine signaling is when there's a signal that's been transmitted within adjacent cells, and it passes through the gap junctions. Okay. So if we look in this image over here, you have cell 1 and cell 2. You will have the blue in the middle, that's the gap junctions. Okay. So the gap junctions is what are connecting one cell to another. Now, with these cells, you have a hemi channel, and the hemi channel is known as a connexon. And what it consists of is 6 proteins, and then you have a hole within the middle, like a pore within the middle. And that is what creates the protein channels. So it's like you've got a channel or a tube passing through it, but you've got 6 types of connecting proteins around it, and that's what forms the pore within the middle. And what it helps or functions to do is to release signaling molecules or ligands such as ATP. ATP is an abbreviation of adenosine triphosphate, which is the energy source used by all cells. So when we say that the cells producing energy, it's the cells are producing energy in the form of ATP. Okay. They also help release small molecules such as glucose. So glucose is a sugar molecule, and that can be uptaken by the cells, and that is what's being used to produce energy via a process called respiration. Looking more closer, now within each of those connecting proteins, you can find 4 membrane spanning domains: M1, M2, M3, M4. You also have loops. There's E1 and E2 extracellular loops, and you also have the cytoplasmic loop. So you can see on one end you have NH2, and the other end it has COOH. Okay. These are the 2 main functional groups that is found within the amino acid structure. The amino acid is the building blocks or the monomers that helps form the proteins. So what are the features of a paracrine signaling pathway? Well, it's local signals, okay, and local levels or concentrations are high. They can be degraded by enzymes or removed by neighboring cells. And the cell that is secreting or producing the ligand can affect target cells only, not itself. And this occurs through a process called diffusion. Okay. So and we're going to discuss what diffusion is later on. So the signals can move by a process called diffusion, and this is via the extracellular matrix, ECM, to allow quick responses. So cytoskeleton consists of 3 types of proteins that help the cell with shape, movement, and organization. The first type is actin filaments. So this is more more round. You also have intermediate filament, which is more a twisted version, and you have microtubules that are thin and long. The ECM forms a meshwork of proteins secreted from cells, and what that does, it helps add strength and support. So you can find these structures here within the diagram present of the animal cell. So you can see the nucleus, which involves controlling the cell, the cytoplasm where chemical reactions take place, and you have the plasma membrane that controls what enters and leaves the cell, and also the 3 types of proteins that form part of the cytoskeleton. Now, looking more closer, you can see that the extracellular matrix over here consists of different proteins such as collagen, elastin. It also has fibronectin and glycoprotein. Glycoprotein is a type of protein that consists of a sugar as well as a protein. And you also have integrin and actin filaments. Now, within the integrin, you can see that it is within the plasma membrane, so it's like a transmembrane. And the cytoskeleton, the first step over here, we have the actin filament there. So this is more of a closer look. So what is diffusion? Diffusion is a type of cellular movement, okay? It's a type of how particles and molecules actually move. And this is from an area of high concentration to a low concentration, down a concentration gradient. So let's have a look at this image here. So extracellular is outside the cell, intracellular is inside the cell. So you can see outside of the cell, there's lots of signaling molecules. So there's a high concentration of the signaling molecules. Now, if we go past the cell membrane and inside the cell, you can see that there's a few there. So when there's a few signaling molecules, we say that there's low concentration. So what happens is the area outside has lots of more signaling molecules. So that is what moves or diffuses through the plasma membrane inside of the cell. And a concentration gradient is the difference between 2 concentrations. So when we say down a concentration gradient, you can see that it doesn't require a lot of energy, okay? Because it's moving from an area where there's lots of signaling molecules to an area that is low concentration. However, when you go against the concentration gradient, that requires energy because you're moving from an area that has low concentration to an area that has high concentration. So let's look at an example of paracrine signaling, and that is neurotransmitters. So what are neurotransmitters? Neurotransmitters are chemical ligands that transmit signals via adjacent cells. And in order to understand the role of the neurotransmitters, we're going to go back to the actual organ system here. Now, the nervous system is the organ system that is found within the body. And it's divided into 2 types of systems. Central nervous system, which comprises of brain and a spinal cord, and a peripheral nervous system, which consists of the nerves. So what's the function or the role of the nervous system? What does it do? It helps communicate, okay, via electrical messages. So electrical messages is otherwise known as electrical impulses. And this is what helps coordinate or to plan out the necessary response to the stimuli, which is the signal. For example, if there is a change in temperature, or if there's a change in taste, or protection from danger. So the central nervous system, we mentioned earlier, consists of the brain and spinal cord. So this is the area that receives the information and makes a decisive point to coordinate the response to the stimuli. The peripheral nervous system, it consists of the connective nerves. So their role is that it connects to the sense organs and it also connects to the effector to conduct a response. So in order for the message to reach the brain and the spinal cord, it requires the peripheral nervous system to deliver it. So here's a close-up image of the nerve cell or the neuron. So they are specialized cells that help transmit their messages electrically. So the dendrites is what receives the electrical messages. And then you have a long tube called the axon, and that is what carries the electrical messages or impulses from one neuron to another. Now, along the axon, you can find these specialized cells called Schwann cells. Now, these Schwann cells, they have a fatty layer called myelin sheath. And what that does, it helps prevent shock electrically or any leaking of the electrical impulses. As a result, it helps provide insulation and speed up transmission of messages. Between each of these Schwann cells, there is a small space known as the nodes of Van Veen. So they allow the electrical impulses to jump to send the messages at a rapid rate along the axon. So in other words, the electrical impulse jumps in order to reach from the start to the finish. The cell body, which is found within the middle there, that is the support center of a large surface area and contains the nucleus. Now please note that there are different types of neurons, and each of them have a specified function that helps to deliver, coordinate response, relay the response, to initiate. The axon terminal, this is finely branched to communicate with other cells. So, as you can see, we have two neurons. So we've got neuron 1 and neuron 2. And you can see there's a small gap between them. And the gap between them is known as the synapse. So the synapse is the gap between one neuron and another. And the reason why there is a small gap there is to allow signals to travel quickly in one direction. But that is done as a chemical way before communicating electrically again. So let's begin. There's an electrical message that has been transmitted. Once it reaches the axon terminal or the ending, it converts into a chemical, and then when it reaches to the dendrites again, it turns back into electrical. So last lesson, I introduced to you one of the ways in how you kind of present your notes within your exercise book, also in terms of revision. So this is more of like a pathway, step-by-step process. So a start button over there, and then you have inputs. So the input information is that the stimuli has been sensed by the sensory organs. So the sensory organs could be hand, where you can actually touch, the ears, where you can hear, the eyes, where you can see, the nose, which you can smell, and also the mouth, which you can taste. Okay. So these are the most common form of the sensory organs there. So once a signal has been received by a branch connection called dendrites, the electrical impulse then goes down the long tube called an axon. When it reaches the axon terminal, it turns from an electrical signal to a chemical signal, and this where the neurotransmitters is required. And these steps continue from one neuron to another until it reaches the chemical nervous system. So in the central nervous system, what you can find is the brain and the spinal cord. So once you make a decisive response, you have the output. So the output is what sends the electrical message via the neurons. And then the whole cycle appears again, where the signal received by the branch connection called dendrites, the electrical impulse is transmitted down a long tube called an axon. The axon terminal transmits the signal as a chemical impulse via neurotransmitters to another cell. Okay, and then the electrical impulse reaches the effector or muscle gland to start a response. So it can either be a gland or a muscle. These are the two main effectors that are being aimed for. So let's look at how the chemical impulse being converted from electrical to chemical, then chemical to electrical again. So let's have a closer look. So at the axon terminal over here, so you've got a presynaptic neuron and a postsynaptic neuron. So there's neurotransmitters. So neurotransmitters, these have been produced by precursors. So if we have a recap in terms of caspases before it's been activated is procaspase. So that's the inactivated version. So similar case with the neurotransmitters, they have the enzymes that produce these neurotransmitters, and then they have an inactive version. So you have like the precursors there before you have the active version, which is the neurotransmitter. And then the neurotransmitter, this is what has been packaged within small packages called vesicles. And this is what is being found over here. So the electrical impulse reaches the presynaptic neuron in the axon terminal. So with the vesicles, they fuse, okay, they stick or they adhere to the membrane of the presynaptic neuron. And this allows the neurotransmitter to be released and then diffuse again across the synapse and binds specifically with its receptor on the postsynaptic neuron of the second neuron. So that is how information has been transmitted. So to prevent overactivation of the neurotransmitter, what you would have to do is to destroy the neurotransmitters once a signal has been reached by enzymes, and this helps control the activation. Another way is that it can be reabsorbed back into the presynaptic neuron. So let's progress on to contact dependence. So contact-dependent signaling involves ligands that bind to the cell surface of another cell via the receptor. And a key example of this would be the plasmodesmata. So the plasmodesmata, they are pores found within the plant cells, and they're filled with water, and what it does, it helps connect the cell membranes of nearby cells. So if you look at the first image over there, you can see that the plasmodesmata is between cells that are very close together. And what that does, it helps to transport material. What type of material are we referring to? Small signaling molecules and ions through specific channels. It also involves within exchange. But that also depends on the size of the actual molecule. Just because it's a signaling molecule, that does not mean that any type of signaling molecule can pass through. It has to be a certain size. So large molecules cannot pass through because of the size, and an example of this is proteins and DNA. Now you can see there's similar structures between the animal cell and the plant cell, and there is additional structures that you can find within the plant cells, such as chloroplasts. So chloroplasts, these are a structure that is green in color, and they depend on the sun. So they absorb all of the sunlight, and this is what helps the plants to make their own food. And the food that they mainly are interested in is glucose. The vacuole, this contains the nutrients or the passage of, for example, amino acids, mineral ions, water, anything that is like a more of a storage. Cell wall. The wall of the cell wall, it helps to support the cell and keep it in shape. We will now refer to the autocrine signaling, which is the third type of signaling pathway. So autocrine signaling, otherwise known as chemical signaling, has a number of key points that characterize it to be considered as an autocrine signaling. So the cell secretes ligands that can bind to their own receptors on its surface. It can affect nearby cells of the same type, and the signals are due to sensitivity of density. So sensitivity means it can sense, and density is in reference to mass and volume. And it often occurs that during the early development of an organism, to ensure cells develop into the correct tissues and induce function, for example, pain or immune response. So these are the key features of autocrine signaling. So let's go through an example. So monocytes, they're a type of immune cell, and the role of the immune system is to help protect the body from infection and disease. And monocytes are actually produced within the bone marrow. So this is the soft, gooey part within the bone. And they produce proteins called cytokines that affect themselves as well as other immune cells. So cytokines, they help facilitate communication. Where there's like a crosstalk between immune cells to initiate a response. And one of the types of the cytokines is interleukins. And today we're going to be specifically talk about a particular interleukin, which is interleukin 1. Now, interleukin 1 is produced in monocytes in response to an external signal. And it can bind to the cell surface receptors on the same cell that produces interleukin-1. So here we have an image of the monocyte there, and we have cytokines, and the cytokine is able to bind to the receptor that is found on its cell surface. So what are the roles of cytokines? They help regulate mitosis for growth and repair. They help regulate differentiation in monocytes to form another type of immune cell called macrophages. So macrophages is a form of a non-specific immune responses because once it sees a foreign material, it just engulfs it and then digests it, rather than specifically recognizing a particular protein on its surface. Let's now look at signaling type 4, which is endocrine signaling. So endocrine signaling requires hormones. So hormones are chemical messengers that help target cells, and they actually reach to the target cells via the blood. So you have the endocrine cell. It secretes or produces the hormones. And these endocrine cells are what is found in structures called glands. And you have a variety. For example, you have pancreas, you also have the pituitary glands, a variety of different types. So when you have an endocrine cell that secretes the hormone, it gets released into the blood and travels to the target cell. Once it reaches to the target cell, you will identify or be aware that there is low concentrations. And this is because it has been diluted. Now, there is a key difference between neurons and also the hormones, because the hormones, they produce a slower response, so it will take time for it to actually reach the message, and it's a long-lasting effect in comparison to neurons. So let's have a recap on the pathways here. Receptor activation, so each hormone can bind to specific receptors that have a complementary shape on the target cells. Then you have a variety of different proteins and enzymes that are involved to help transmit this signal through the cell. And you also have cellular response. So this can be either regulatory genes, can alter, for example, the enzyme or the protein. And there's a variety of different types of hormones. So steroid hormone, they are made from fats or lipids by the cholesterol. They are soluble, so they can dissolve through and pass through the membrane. They do enter the cell and they can act on the DNA. And a key example of estrogen. So estrogen is a hormone that is involved in the lady's menstruation, so the period. It's also involved in pregnancy. It's also involved when a girl starts to develop growth. So any secondary characteristics are actually being facilitated by the estrogen. Another example is eicosanoids. So these are lipids, which is fats, and it's produced from the fatty acid chains of the phospholipids found within the plasma membrane. So if you look at reflected in every structure of the cell membrane we've looked at, you can see there's a circle, and then you've got like a kind of a two fork. Okay, now the circle there, that's the phosphate, and then the two tails that are bottom, that's the fatty acid tails. Okay, so the lipids are produced from the fatty acid tails of the phospholipids, and that is found within the plasma membrane. A key example of this is prostaglandins. The prostaglandins also has an effect on the female reproductive system. So, for example, it's involved in the lining of the womb. Okay, it's involved in changing the blood vessels during early pregnancy. It can help increase the contraction or the movements of the myocardium, the myometrium, sorry. So the myometrium, this is the muscle layer that is found within the womb. So these are some of the functions of prostaglandins. Now they have the amino acid derived. So amino acids, as you mentioned, they're the building blocks that make the proteins, and how they join together is via peptide bonds. So example of this is adrenaline and noradrenaline. And with these examples, the amino acid that helps produce adrenaline and noradrenaline is tyrosine. And the effects of adrenaline and noradrenaline is a fight or flight response, is emergency situations. So you could have your eyes being dilated or widened. You can have an increase in heart rate. You can have your blood vessels, instead of becoming larger, they become more thinner, so it's more constricted. It helps produce more glucose in response to help with the energy. Yep. So these are some of the functions of adrenaline and noradrenaline. And the protein hormones. So example of that would be insulin. So insulin is a type of protein hormone that is being released when there is high levels of glucose in the blood. So when that is actually being released, it helps convert the excess or the addition glucose and helps store it in as glycogen. Now, they are not soluble in the membrane, and they do not enter the cell. By the end of this lecture, you should understand that cell-to-cell communication consists of direct, paracrine, contact-dependent, autocrine, and endocrine signaling pathways. Cell signaling consists of 3 steps: receptor activation, signal transduction, and cellular response. Dysregulation of cellular signal transduction pathways underlies most of the hallmarks of cancer. There are different types of signaling pathways and vary based on the distance traveled to reach the target cell. Protein-based ligands are insoluble in the membrane and does not enter the cell. Fat-based ligands are soluble and pass through the membrane. There are key differences between nervous system and endocrine system, and there are also similarities between them. So they all involve a signal being transduced. They also have an organ being involved. They also involve a response. Okay, but they vary in how the message is being sent across, in the method and the length and how long does the response take. So within the nervous system, the information is being transmitted electrically, okay, across the neuron. And The target organs is actually localized cell, so it's from one cell to another of the nerves, so the neurons. The type of response is involuntary or automated, or it can be voluntary. The length of the response is short, and the duration or the time is quick. The endocrine system, the information is transmitted chemically as hormones, and the hormones are released into the blood. And it mainly targets cells that are distant, so many cells are affected, and it's more of an automated. So for example, if there is a glucose or sugar level that is low or whether it's high, the necessary hormone is being released. And the length of the response is long, and the duration of the response is slow. So here you can find a variety of different references to help increase or extend your learning. Thank you all for listening. I hope you have benefited from this lecture. Please feel free to email me if you have any questions. You have the glossary to support your understanding for any of the keywords. You also have a quiz to assess your learning. You have the HN designs, which consist of simplified and basic images. And hopefully next week we'll begin with step 1, which is receptor activation. And I look forward to sharing information with you. Take care. Bye.